partner, Sarah, to introduce our speaker for today, Swaine Chen. It's a pleasure to have him here. So I'll hand it over to you guys.
Hi, good morning, everyone. Thank you for joining us, especially slightly earlier this morning. And we'd like to say hi also to our first speaker of the year, Swaine Chen. And I think our speaker, I'll just do a little bit of an introduction to him before you guys get to know him a little bit better. He is an associate professor of medicine in NUS. And in addition, he's also a group leader at the Genome Institute of Singapore. So his research combines his training in medicine and science to understand a little bit about why bacteria are able to cause infections in human. So if you guys are wondering, today we'll be exploring the promise of science at the intersection of data, research, and creativity. Hi, good morning, Swaine. How are you today?
Good morning, Sarah. And thanks for having me on. Thanks for giving me the chance to come on. I've heard about this series for quite some time, so I'm really excited to be here.
Yes, we're so excited to have you this morning as well. Maybe we'll start with, could you tell us a little bit about yourself and maybe just a little bit more about your role in NUS for everybody to get to know you a bit better?
Sure.
Yeah.
So my name is Swaine, and as you may be able to tell from my accent, I'm American. I was born and raised in the US, did all of my education there, and was on a pretty academic path from the beginning. So I, sort of took lots of tests and ended up going to medical school, also got a PhD in biology. And that path then took me, down the sort of standard academic path where I, after my PhD, I did some more training and then became an assistant professor. And that was when I moved to Singapore to start my lab, my own research group as a professor. And that was at NUS about 10 years ago. And since then I've been working, and just as you said, the main thing that we focus on is trying to, combine the training that I've had in the past, both in medicine, as well as some of the other different sciences, including mathematics, to really understand how it is that bacteria cause disease in humans. So there are lots of bacteria out there, and not all of them cause disease. And the general approach that we take is to use a genetic approach. What that means is looking at the DNA, right? So you may sort of hopefully remember at some point in school, they told you that all organisms have DNA. It's like the genetic code, right? Or sometimes people call the genetic script. And one of the things, and I think like the way this fits into the theme of promise, is we have a lot of these books, a lot of the genetic books out there, but we don't know how to read them. Yeah. And this is what we're trying to do at my lab, to try to understand what it is about this DNA that makes these guys able to cause disease.
That's so interesting. And I think it's such an interesting topic that we'll be exploring today. I know this is a little bit of a tough question, but what can you tell us about science and creativity, which I think maybe people don't really put them together, but I believe that is a strong part of your kind of what you do every day, right? And maybe tell us a little bit on how does that connect with you every day at work?
Yeah, so science and creativity, I would say one of the most immediate things that comes to mind when you put those two words together, we are taught, when I'm talking to graduate students in my lab or other trainees, I am often trying to tell them that within what we do have to be very creative. We have to kind of imagine what the problem might be. A common thing that I'll say to them is, if you can't think of what the idea is, right? So say we have some problem with some experiment and the experiment didn't work. And they're trying to troubleshoot, okay, well, did I switch something? Did I add too much of one thing or another? And as soon as you have this idea, then you can go and try it and fix it and see if that really sort of, if that was really the problem. But if you never stumble on the right problem, if you don't imagine what that right problem is, then you're just never gonna fix it. And so that might sound a little bit trivial at the point of, we're just sort of following some recipes and doing this. But when we're actually trying to understand, how is it that some new pathogen, right? And this is, I think, on everybody's mind these days, right? We have these things that cause disease. We've never seen them before. And they may use different ways to cause disease from things that we've seen in the past. And that's really where a lot of the imagination comes from. Yeah.
I think that's true. I think nature is very, very creative in the way that they are constantly changing and adapting to things. just going back on our theme of promise and what it means to, I think every chapter interpreted differently, and we'll be very interested to know on why did you choose to speak on the theme of promise, and how does this actually relate to your field of study?
Yeah, so I mentioned that the general approach that we use in my lab is a genetic approach, right? Looking at DNA. And maybe if I can share a slide just to go over a couple of ideas here.
All right.
So let's see here. One second. Okay, so this image, Science and Nature, these are two of the sort of most famous academic journals where, you know, this is sort of the standard thing. We have to do research and we publish it. And both of these, their cover image at this time, you can see if you look closely, the date is 2001. So it's about the human genome, right? So this is that kind of book of life, right? That genetic script. And this is the thing that we can now determine what it is, but we can't really read it. Okay, and this was very clear at the time. So one thing that was, Bill Clinton was the president of the US at the time. And he said, you know, genome science, right? So genome being the entire set of DNA. Genome science will have a real impact, right? And even more in the lives of our children. Okay, and so this is what everyone was saying. This is why we need to do this. This is what science is going to do. It's going to revolutionize diagnosis, prevention, and treatment of most, if not all, human diseases, right? And then we'll be able to cure diseases like Alzheimer's, Parkinson's, right? Things that we hear about and are scary. I mean, or maybe you don't hear about them. Most people know people that have had, either have experienced themselves or know people that have experienced with these diseases. These are not good. And we want to be able to cure these by attacking their genetic roots. Okay, so this quote's from 2000, and I think that really set the stage for, you know, why genomics, studying genomes, this was the promise that we were supposed to bring. And that's sort of how I interpreted promise, and why I think genomics is a really natural topic. Genomics and science is a natural topic for this. If you fast forward 10 years, so there's, this was this article published by Nicholas Wade. So 10 years later, this is what he said, right? The human genome, like medicine has yet to see any large part of the promised benefits. Okay, so that primary goal, which was to get at the genetic roots of common diseases, remains largely elusive, right? And then, you know, this is particularly something you know, when scientists hear this, it's like, this stings a little bit, right? After 10 years of effort, we're almost back to square one and knowing where to look for the roots of common disease. Okay, that's a bit tough to hear.
After 10 years,
So that's the promise, right? And so let me show one other slide, which relates to promise here as well. So another way we think about this, and another reason why this is so exciting, is, you know, if you guys think back 10 years ago, before, or maybe 15 years ago, before smartphones existed, how much has your life changed?
Yes, definitely.
Right? I mean, it's gone from something that you've never even heard of to something that people will carry 2 or 3 of these around at all times, and you can't leave the house without it. And, you know, those smartphones, so the iPhone came out in 2007. In 40 years, we took computers from things that would be like the size of a small car or even the size of a building. And in 40 years, the progress in computer technology brought this down so that you have even more power than this entire room up here. You have more power in the palm of your hand, okay? And so if we look at what happened in genomics, well, in 2010, when and this is me over here for a sizing scale, this is a machine behind here, right? And again, it's like the size of a small car. This was a genome sequencing machine. It was one of the machines that we would use to actually get those DNA sequences that, you know, then we want to try to interpret to find those genetic roots of disease. Okay, so we bought that machine, or that machine was available in 2010. We bought it shortly afterwards. And then I'm holding in my hand another device, which was bought in 2014. And that device in my hand that device in my hand does almost as much as that machine behind me. Okay, so in 4 years in genomics, we kind of did what it took 40 years in computers to do, just in terms of like shrinking the size down. Okay, and I think, you know, one of the things that we've always talked about is the reason that matters, the reason, like, size doesn't really matter, does it? I mean, the computer's a computer, but the reason that matters is if you can hold a computer in your hand, all of a sudden the applications or the integration with your life can be totally different.
Yep.
Right? It's no longer the realm of big companies or governments that have computers. Everybody's got a computer. And that's sort of one of the things that we think about genomics as well. Okay, and so just the last graph I want to show, Moore's law is that top line, that's an exponential rate of increase, or it's an exponential decrease in cost for computing power. And that's been going on for decades. And that exponential rate is what has given us that 40-year kind of shrinkage, where now we can all carry cell phones that are very powerful just in our pockets. And if you look at the other graph, the one with the dots there, that's what the progress that genomics has made. And obviously, as it goes down faster, that means the cost is going even faster, right? Or the cost is going down faster. So genomics has gone even faster than computers, which is kind of the gold standard for super rapid progress, right? Genomics has gone even faster. And so you put these two things together, right? That genomics has gone so fast, the form factor has gone down, we have the technological capability possibly to integrate this into our daily lives and have that same kind of transformation or even more transformation than computers have. And yet there's this feeling that after 10 years, we're back to square one and knowing where to look. Okay, so so this is where it is. And you know, I think then the next step would be to say that a lot of what this is, and you referred to this already, being able to drop the cost, this is really an aspect of data acquisition. We have all the genetic books. We still don't know how to read them, right? So now we've got this huge library, and really the problem is, can we read and understand what's going on?
Yeah, I think that's quite interesting that by, you know, what you're sharing with us is that in the span of just a few years, we do make a lot of breakthrough, but sometimes at the same time, we also realize that, you know, we are kind of like, we still have a long ways to go in terms of like questions and where we should be in the next few years in terms of the field of genomics, right? Just going back to data and creating breakthrough, right? And talking about something that I think is on everyone's mind right now, which is the current COVID situation. Maybe could you help us elaborate a little bit more on the maybe vaccine, and again, the creativity behind the research and kind of like what you mentioned, right? Just identifying what is the problem and looking at it from a creative angle. Maybe could you share a little bit about that with us?
Yeah, sure.
On your perspective,
so,
maybe.
You know, maybe we'll jump back to another slide here, if that's okay.
All right.
It helps us talk about this. So, okay, I'm going to show this. All right. So everybody, you know, it's all over the news and you can't avoid this, right? So we have COVID-19, it's caused by this virus called SARS-CoV-2. And you'll see this in the newspapers. Most people have never really sort of tracked a vaccine development, right? But now one of the amazing things is everyone wants to know when this vaccine is coming. And so here's this really nice graphic that was made to show just how amazingly fast it was for us to make the SARS-CoV-2 vaccine. So SARS-CoV-2 is this dot right here, and this red and blue dot is because we discovered that SARS-CoV-2 was causing COVID in 2020, and it's in the same year that we had the vaccine. And that's basically never happened before. Okay, so here's a few other diseases on the y-axis that you might have heard of, right? And these things sometimes, you know, you look at this typhoid fever, right? It's over 100 years that it took us to develop the vaccine. So, you know, this is one of the things that gets trumped in the news a lot. And then the thing that you don't realize is that the mRNA vaccines, or you might realize that there's a different technology, and some of the stores are picking this up. But to get to the point where we understood enough to use an mRNA technology to make a vaccine, Like, so here's another paper that was published not too long ago, which went through the timeline of the key things that eventually built up to us being able to use this mRNA vaccine, which is down here in 2020. And this graph would go from, on the top, it goes from left to right, and then it loops back and goes from right to left down here. And, you know, if I can just point out a couple of things here. So the initial concept, let's see, maybe let's try this. The initial concept was right here in 1989. Okay, that you could use, we understood what mRNA was. So mRNA is just this molecule that's inside cells. It's absolutely required for life. And almost, you know, all cells have this. But the ability to understand enough of how it worked and then to say, hey, maybe we could use that to make drugs or vaccines. We had that idea in 1989. And then, the idea of drugs and then vaccines in 1990. And basically all of these other boxes here, we're trying to realize that dream, trying to say, can we actually use these mRNAs for a vaccine? And basically it never worked. And the thing that you learn, of course, or the thing you hope you learn is, why did it not work? And what you realize is there's all these other things that have to happen. One of the more obvious things is that mRNA, because it's a natural molecule, so our cells know how to make mRNA, but they also know how to degrade it and recycle it. And that was one of the big problems. How do you stop these from getting degraded when you inject them into people? Like, they need to last long enough in order to induce that vaccine response or induce that immune response. And so it took a lot of additional technologies that had to get put together or invented to try to overcome these issues. So sure, like the initial idea was 30 some years ago, and that's a clear idea, and that's still the idea that we're using today, right? But it took 30 years to get there, to fix all these other problems. And those are problems that you could never, or people basically had a lot of trouble imagining along the way, right? You had to see the failure, like you know, it would go into human trials or animal trials, and you would see that it just didn't work. And then you'd have to spend a few years to figure out, why did this not work? And that's where the creativity comes in, right? I mean, there's some data we can collect and try to look, but then you have to understand what the problem is and then try to find a solution that's compatible with injecting things into humans. Okay, so hopefully that makes sense, like in terms of like trying to think about this. I think one of the really exciting things, the last thing I want to highlight on this slide is, if you look at typhoid fever, or you know, things like polio and mumps, all these other diseases you've heard of vaccines. One of the reasons people are so excited about the mRNA vaccines, and the reason that this dot is drawn here is just on the same year. It's not drawn as a 30-year process, right? It's drawn on the same year. Is that, you know, to vaccinate the original polio vaccines, you actually had to have the polio virus to make that vaccine. Okay, and then you had to grow up the virus, and then you would actually inject an activated virus or sort of crippled virus into people. And, you know, so if you now want to make a mumps vaccine, well, that polio virus doesn't really help you, right? Mumps is a totally different thing. And so what you might say is that, you know, you just sort of, well, okay, the idea is that it's not programmable. Like, polio is not a platform. The vaccine that we use, the way that we did that, it's not really a platform that we can generalize. But for mRNA, okay, because mRNA is like this genetic code as well. It's related very, very strongly back to DNA. It's just something that it's almost like typing on a keyboard, right? We can just switch around the characters. It's what we would say programmable. So now that we've solved all of these 30 years of problems, all the delivery technology, right? The safety issues, making sure it doesn't get degraded. Actually, at the end of the day, the mRNA, we can just type on a keyboard, and now that mRNA matches SARS-CoV-2. Or we change a couple of characters here, and now that mRNA will match new variants of SARS-CoV-2. Or we change a few other characters, and maybe that variant now, maybe it'll match to the Zika virus instead, right? And so those things, that's really one of the super exciting things. And I would say it's not just in vaccines, but this super exciting idea that we can continue to leverage on those 30 years of discoveries, right, to really accelerate future development of new vaccines and keep all of those vaccines down to less than 1 year. That's the big promise. And there's other things that we can do. So just to give you one more example. So there's this outbreak. Let me see, clear these guys. There was this outbreak in 2015 in Singapore with this bacteria called Group B streptococcus. And so this was associated with the consumption of raw fish. One of the things that was very difficult about this outbreak was no one had ever seen this disease before, right? So just like COVID, no one had ever seen this disease before last year. And what we did in my labs, we helped to do some of the DNA sequencing, some of the genomics, right? Looking at the that sort of genetic code inside that bacteria, and we were able to establish that the bacteria causing disease in humans was exactly the same as the one that was found in the food, and that established a strong link. Okay, but in reality, when were we able to do that? So if we go back and look at the data, the outbreak started probably 6 months before we were able to figure out what was going on. Okay, and so just like the vaccine things where we've got all this technology, and now in the future we can say, oh, now that we see something new, we can really go very quickly and develop a new vaccine. One of the other things that's related to that conceptually is in genomics, we can see, based on how this happened, we can see that we could probably detect the outbreak over here in February or March. Now, now that we sort of know what happened. But the idea about genomics is, again, it's a platform. And if we put those systems in place, it doesn't matter what the new outbreak is. It doesn't have to be something we've seen before. We actually could detect outbreaks. It's a general process, a general platform to detect outbreaks no matter what's causing them. And we could detect it over here in the February, March range, where just the first few cases, and we can already see something's happening. Okay, so that's sort of why I think, like, really the data and these platforms and all the things that we've been working on for so many years, this is really kind of the promise that we're trying to get past the statement of, like, we're still back at square one. Okay.
Yes,
does that make sense?
Sharing that. Yeah, I think that's quite fascinating, to be honest, to hear you speak about all of this, and it's so elaborate. Like, I feel like it's really helpful to also see a few images of you explaining. I'd love to actually pass it to the audience. I feel like people do have some questions related to your talk on Promise, but also about, actually the field that you're invested in, genomics. So I'll pass it on to my colleague Bryce over here, who will take Q&A from everyone.
All right. Hi, good morning, everyone. Can I just ask if anyone has any questions? Feel free to put it in the chat, or you can also unmute yourself and ask Swaine yourself directly.
Looks like there's one in the chat already.
Yes.
So maybe we'll just take this. So Ivan, do you want to unmute and we can just sort of talk, or I can just read it out if you don't want to do that? Okay, maybe I'll just read it out. So Ivan's question is, what are some of the key ethical challenges in genomics, and how do we manage them and move forward? And so this is a question we get a lot. This is a question that can be very sticky. And actually, I really appreciate how you asked two questions, because it's clear that I can't just give you an answer to tell you how ethics should be for genomics, right? This is not a simple issue at all. And it's going to keep changing as technology develops and we're able to do different things. And so I really like how you have that second question, how do we manage them and move forward? Because I think that's really the only answer we can give, right? That it's not what are the ethics, like that's not, that's sort of, the problem is much bigger than that. What we need is, like, how do we deal with this, and how do we keep up with the technology as it's changing to sort of figure out what the ethics are. And so you're absolutely correct. I think, maybe we won't say too much about, like, what the actual kind of issues are. There are influences of genetics on disease. And, I think one of the first things that comes up is, like, the insurance companies use that. And I'm actually really happy to say that Singapore recently put in a rule to place a moratorium on the use of genetic data for insurance purposes. So that actually just was announced in the last week or two, right? To sort of kind of put that issue at rest, at least temporarily. So there is awareness of this at all levels, and not just with scientists, but with the lay public as well as the government. And then to get to the question, like the real question was, how do we manage them and move forward? And I think, this is one thing I really appreciate everyone being here, and you guys give me a chance to talk about this, because I think one of the ways that is not a good way to deal with this is to try to just have all of our little silos, and I do this and you do that, right? So I study genomics and I do this, and I don't need to care about ethics because I'm just the scientist over here that does the genomics. And then, okay, I'm an ethicist over here and you do all your science stuff. I don't need to know all this science stuff because I'm an ethicist and I'm going to tell you, like, or I'm going to sort of think about these moral questions. And I think that's really, there's been a tradition of kind of this field specificity that is not so great. And I don't think that's a good way forward. The better way forward is that these technologies just need to be well understood by everybody, because these questions are a societal question, right? These questions are not just the questions for the ethicists. There's not just a question for government, right? It actually affects us. It affects us as scientists in terms of what we can do and what we should do. And so we also can't just sort of say, oh, we're not going to pay attention to politics and not going to pay attention to ethics. I think there has to be an effective crossover between all these different fields. And that's what I would say is the main thing we need to do to manage. And that's the more healthy way to move forward, that then we can actually have meaningful conversations. Certainly there's a specialization and expertise, but we need to be able to have meaningful conversations across those in order to manage these issues and make them relevant to technology. Okay, so hopefully, Ivan, hopefully that makes sense.
Right. Thank you so much, Swaine, for answering Ivan's question. I think we got another one from.
Yep. So we have another one.
Yeah.
So Ree, do you want to unmute and say this, or I can read it out again? Okay, maybe I will read it. So the question is, what would you say is a promise you'd like to deliver on or contribute to your field of science? So, it's another big question. And I would really go back to that article, that quote from 2010. Okay, so at that time, I wasn't running a lab, and I was in genomics, but not quite in the same thing that they were talking about. But this is a quote that I kind of use, and I remember I use it in my teaching a lot, because I think it really sort of cuts to the heart of the matter, right? That after 10 years, we're back at square one, right? And so the promise that I'd like to deliver on is to make sure that that's not what people think about what we've done 10 years on, 10 years later, right? What are the actual contributions? And I would, to add just a little bit more flavor to that, I mentioned this particular disease, this group B streptococcus, okay? And so that's one disease, that's one bacteria that causes a disease. It was a new disease, so we never knew about it. So that's cool scientifically, but also important in terms of, now when patients come in, we can say, oh, well, this is another possibility, instead of just, I don't know, and who knows what happened. Okay. And so we're working on that disease and trying to understand how is it that group B streptococcus, why do we not see this fish-associated disease before? Why was this a new disease in 2015, right? Even with all this technology we have. Okay, and so that's a big question, and we can say, all right, if we answer that question, now we understand something about group B streptococcus. So that's one thing that we would like to do. But the bigger thing we'd like to do is actually that general system, right? That's the real contribution I want to do. Is like, I don't want to just study one disease. I mean, we have to study individual things. We have to study specific things. But in so doing, the hope is that we have general systems that then we can detect any outbreak at the beginning, regardless of what's happening. So that when the next new disease comes along, it doesn't take us 3 to 6 months. It takes us 3 to 6 days. And that's exactly the same kind of thing that is happening with that mRNA vaccine. So we can make vaccine after vaccine after vaccine. We can use the inactivated viruses, which have been used for decades and decades. But now we got to the point where they've developed all the technology that they can make a new vaccine in 1 year. And it's not a new vaccine only for SARS-CoV-2, right? The promise there is that you could, the next virus that comes along or the next disease that comes along, in principle, we could also do this in 1 year for that new thing as well, right? So that, I think, you know, by studying these particular systems, but then having that general impact or that platform that then really allows us to accelerate everything, that's the kind of contribution that I would like to make. And that certainly is not something that's going to happen this year. Hopefully on a 10-year time scale, that's sort of more reasonable for us to do.
Well, thank you so much, Swaine, for sharing your promise and what you'd like to contribute to this world through your work and also your research. So for now, maybe I'll pass it back to Sarah and Luana.
Yep. So we just like to thank you again, Swaine, for joining us this morning and sharing with us, I think, quite an in-depth view of what you do at work and how actually it relates back to creativity and our theme of promise. I think it's quite inspiring for us to kind of head into our weekend to know that, you know, we do make progress in this world in terms of facing this current pandemic and this current time. We just want to invite you to maybe take a picture with us, like a virtual screenshot, if you will. We like to invite everybody also, if you're okay, to maybe share your screen so that we can take a picture of all of you guys. You can have a virtual background like us. Then we'll just take a quick snapshot of everyone who is here this morning. I think that would be quite a nice way to start our year.
Sounds good. Thank you guys for having me on. It's always fun to talk about this stuff, especially with an engaged and curious audience.
Yes. All right. We'll just give you guys a little bit of time to maybe turn on your camera. And then I'll take a quick screenshot of everyone.
Hello,
Hi, so nice to see everyone's faces. Hopefully we'll be back into our regular physical space soon so we can see everyone again. Hello.
I think next time we start with the photo. We start the talk with the photo. Everyone's faces in the morning.
Yeah.
Thanks for tuning in, guys.
That's right, with coffee mugs right here.
Yeah.
Yes, we love our coffee mugs.
Hello, everybody. It's great to see the cameras coming on.
All right. Okay. So maybe just say smile. Good morning. Okay. All right. I'm taking a picture in 1, 2, 3, smile. Okay, let me take one more. Okay, 1, 2, 3, smile. Okay, thank you everyone, and thank you for sharing your faces with us. Have a great morning, and thank you so much again, Swaine, for inspiring us. Thank you.
Thank you, Swaine.
Thanks, Luana, Sarah.
Bye-bye. Have a great weekend ahead. Bye-bye.
Bye.
Bye. Thanks.
Bye.